all, let's continue to the next series of nutritional biochemistry, that is carbohydrate episode 2. What are the important properties of monosaccharides? These monosaccharides form a water-soluble, colorless, odorless, sweet crystals, and they show a specific reaction to aldehyde or ketone and hydroxyl groups. Let's see about the important reactions of monosaccharides. The first one is esterification. This ester in the sense they form hydroxyl group with acid. This esterification is the process in which monosaccharide that have hydroxyl group form esters with acids. From the picture, carboxylic acid and alcohol combine to form, in presence of hydrolysis, we will get esters and water. These esters form a fruitful flavor. That is, in jackfruit, it is said that about 13 type of esters are present for the particular flavor of that fruit. The next important one is oxidation reaction. While using mild oxidizing agents such as sodium hypoiodate, bromine water, which oxidizes aldoses to aldonic acid. That is, aldehyde group is converted into acid group, that is carboxylic acid group. To get, in case of glucose, it is oxidized to gluconic acid. When we use much higher oxidizing agents like nitric acid, this glucose is oxidized to glucaric acid. And ketose does not undergo this oxidation as the hydroxyl group is not present in that particular compound. Now let's see the formation of a uronic acid, that is dilute hydrochloric acid oxidized to terminal CH2OH group to form uronic acid. In case of glucose, it is oxidized to glucuronic acid. One of the important tests for identification of a monosaccharide is Felling's test. In this, free group, that is aldehyde or ketone, reduces cupric ion in alkaline condition that leads to formation of cuprous oxide, that is Cu2O. That leads to the formation of a red precipitate, that is the indication of presence of monosaccharide. In alkaline condition, that is basic solution, carbohydrate react with oxidizing agent to form red colored precipitate, which leads to the identification of reducing sugars. Then what is reducing sugar? They have either free aldehyde or a ketone group in their structure. In alkaline condition, all monosaccharides, that is aldoses and ketoses, are reducing sugars. And for example, simple sugars, namely glucose, fructose, galactose, can act as a reducing agent as they have a free aldehyde or ketone group that is oxidized to carboxylic acid to form red colored precipitate. The next is a reduction reaction. This aldehyde and ketone groups are reduced to polyhydroxy alcohol in the presence of sodium borohydrate and sodium amalgam, and the glucose is readily reduced to glucitol, that is also known as sorbitol. And these sugar alcohols are also called as polyhydroxy alcohols as they are hydrogenated to form aldoses or ketoses. The glucitol is also same linear reaction structure as chain form of glucose. This glucitol have a same linear structure as chain form of glucose, but the aldehyde group is replaced with CH2OH group at first carbon. These sugar alcohols are also used in low calorie or sugar-free products. And these sugar alcohols are also known as polyols, which is used as a sweetness and bulking agent. They can naturally occur in food that come from plant products like fruits and berries, and it is mainly used as a sugar substitute as they provide very few calories than regular sugar. And let's see more in detail about reducing versus non-reducing sugar. These reducing sugars are carbohydrate that act as a reducing agent due to the presence of aldehyde or ketone group present in the structure of that particular compound. In case of non-reducing sugar, they do not have a reducing agent due to the absence of aldehyde group or free ketone group. This reducing sugar acts as a very good reducing agent. In case of non-reducing sugar, they does not act as a reducing agent. Then this reducing agent also have a free aldehyde or ketone group as we have already discussed. Then they give a positive reaction towards Felling's test. That is, they turn from blue to red colored precipitate. They include all the monosaccharides and some of the disaccharides. In case of non-reducing sugar, they do not react with Felling's test and include some disaccharide and all polysaccharides in non-reducing sugar. A few examples for reducing sugars are monosaccharides, all the monosaccharides, and some disaccharides include maltose and lactose. For example, for non-reducing sugars, disaccharides and oligosaccharides like sucrose, raffinose, and stachyose. Next is one of the important reactions in monosaccharide are ozone reaction. When a monosaccharide is heated with the phenyl hydrazine in presence of acetic acid, the carbon group of monosaccharide react with this phenyl hydrazine leads to the formation of ozone. This ozone is identified by the yellow or orange microscopic crystals with specific melting point. Next one is furfural formation. When this monosaccharide treated with concentrated mineral acids, they form a class of compound called furfural. The organic compound with formula C4H3OCHO forms furfural. It is a colorless liquid that commercial samples are seen in brown in color, which is mainly used for making ink, plastics, antacids, adhesives, nematicides, fungicides, fertilizers, and all. Moving on to the next category, oligosaccharides. These oligosaccharides contain 2 to 9 monosaccharide units that are joined together by glycosidic linkage. Again, these oligosaccharides are classified into disaccharide, trisaccharide, tetra, and all. Disaccharide in the sense, two monosaccharide units or molecules are joined together by the presence of glycosidic linkage. A few examples of disaccharides are maltose by the formation of two glucose molecule, then lactose with a combination of glucose and galactose, and most important one is sucrose, that is also called as table sugar by the combination of glucose and fructose. Now let's see what is glycosidic linkage. It is a chemical bond that is a kind of covalent bond that connects carbohydrate molecule to another group. It might be either carbohydrate or not. Examples of this glycosidic linkage include cellulose on woods of plant, chitin found in cell wall of mushroom, then DNA also contain glycosidic linkage. This bond is formed through a reaction between hydroxyl group on the anomeric carbon of one unit with hydroxyl group of another compound. These disaccharides with free anomeric carbon atom like monosaccharides exhibit reduction reaction with carbonyl group that also exhibit the presence in Felling's reagent and Benedict reagent. Then we have already discussed that the sugars that reduce above reagents are called as reducing sugars. Sucrose is a kind of non-reducing sugar as it does not contain free anomeric carbon atom. And the sucrose consists of two types of sugars, that is glucose and fructose. It's also called as alpha D glucose and beta D fructose. Maltose consists of two alpha D glucose compounds. In case of lactose, alpha or beta D glucose and beta D galactose. Now let's see about the major classification called polysaccharides. These polysaccharides are formed by many monosaccharide units combined together by the glycosidic linkage. Again, these polysaccharides are classified into homopolysaccharide and heteropolysaccharide. This homopolysaccharide in the sense, from the name it is understood that they have only one type of monosaccharide. Example, starch and cellulose. In case of heteropolysaccharide, contain more than one monosaccharide combined to form heteropolysaccharides or heteroglycans. Example, hyaluronic acid and heparin. Now let's discuss about homopolysaccharide called starch. It is a plant polysaccharide also known as amylum. This polycarbohydrate composed of numerous glucose units joined together by glycosidic bond. These starch are produced by the most green plants as the source of energy, and it is stored as starch in plants. The major sources of starch in carbohydrate are namely wheat, potatoes, corn, cassava, etc. The starch is mainly classified into two types of molecules, amylose and amylopectin. While amylose is a linear chain of a glucose molecule and amylopectin is a branched molecule. This structure of a starch affect the digestibility as the linear chain of molecule is easy to break and they impact on blood glucose level. About 65 to 85 percentage of starch consists of amylopectin and 15 to 35% is amylose. This amylose is formed by the straight chain structure, that is include 250 to 300 glucose residues that are linked together by alpha 1,4-glycosidic linkage. In case of amylopectin, it is a kind of branched polymer of glucose that the branching occurs at interval of 24 to 30 glucose residues. This picture depicts amylose, that is a linear structure, and amylopectin branched structure. The glucose units of a main chain in amylose are joined together by alpha 1,4-glycosidic linkage, and the glucose units at a branched amylopectin are joined together by alpha 1,6-glycosidic linkage to the main chain. This amylopectin consists of 3,000 to 6,000 glucose residues. And starch is not soluble in water and they form colloids in aqueous medium called micelles. Then how the presence of starch is identified? It is mainly identified by the iodine reagent. That is, the iodine reacts with both amylose and amylopectin to give a violet color complex. When starch reacts with iodine, they form blue color complex that dissociate on heating and reforms again after cooling. Let's see more in detail about cellulose. It is one of the main constituent of plant cell walls, and it is the main constituent of plant cell wall and most common and abundant of a D-glucose polymer. It is a kind of homopolymer like starch, but difference in chemical structure, which is not hydrolyzed by the enzyme amylase. Then they need strong hydrochloric acid to hydrolyze cellulose to glucose. Glycogen is the storage polysaccharide that is found in muscles and liver of animal and humans. That is, the carbohydrate we consume is digested and excess will be stored as glycogen in muscles and liver. It is a branched polymer with 8 to 10 glucose unit in each of the branch. Like amylopectin, the straight chains are formed by alpha 1,4-glycosidic linkage and alpha 1,6-glycosidic linkage exist at the branch points. And it is said that about 5,000 to 10,000 glucose units are present in glycogen. It is a kind of non-reducing sugar, easily soluble in water, which give a red color with iodine test. Next one is dextrin. They are polymers of D-glucose which are held together by alpha 1,4-glycosidic linkage. And just note that these dextrins are formed due to the partial hydrolysis of starch by the enzymes like salivary amylase, then dilute mineral acids or heat. They form consistency of sticky solution in water, which is frequently used as adhesives. That is example on postage stamps. These dextrins are also used as a thickener or preservative in food. Also, it is found in tea concentrates, caramel, frozen dairy products, low-fat or non-fat products, and hard candies. On hydrolyzing dextrins, we will get maltose, then finally to glucose. The next is inulin. It is a kind of polysaccharide that are made of fructose, that is fruit sugar. They are soluble in warm water, which does not give any color with iodine. They have a sweet in taste and found in vegetables and fruits like onion, leeks, garlic, banana, asparagus, chicory, etc. This inulin is also considered as a dietary fiber as they helps in gut health, then reducing diabetes, weight loss, etc. So far we have discussed about carbohydrate, its classification properties, then monosaccharide, disaccharides and polysaccharide and all. Meet in the next series of classes. Thank you for watching. Do like, share and subscribe.